the fractional abundances of the tumor cells and non-tumor cells in all the other patients. And they have also like distinguished the transformed and non-transformed populations using the chromosome 10 and chromosome 10 deletion and chromosome 7 amplification data. So this is how they are distinguishing that how the acute slice cultures are showing a better picture of the glioblastoma tissue. Like right down there, they have used a particular model to use the gene. They have used the gene signatures to differentiate the populations, the cell populations that are there in the tumor subpopulation. So this is how they distinguished. And what we saw again was, and this is the like the NCBI bio project where we got the like sequence data and all the publication information and everything. And this is the point where Urja has helped me with understanding the marker genes that I might have missed and failed to understand. So I will want Urja to speak about what she wants to present today and how she has like used the like understood the marker genes that were used in the publication. So Urja, can you take over from here?
Sure, sure. Firstly, I'd like to thank you for letting me collaborate with you. It's a wonderful topic. So I have very recently joined this project with her, and from what I've understood, I've been helping her with the project. So what the study has basically done is analyzed the tumor and the tumor microenvironment from glioblastoma patients. So using data analysis and RNA-seq pipelines, they were able to give us gene expression within the tumor populations and the microenvironment. So as Chandrani already pointed out, that they had checked for transformed and non-transformed cell populations, which were then compared to references. So using their RNA-seq pipeline and data analysis, as presented on the slide, there were certain genes that were significantly upregulated or downregulated. So, and they had used actually they used 6 types of drugs, but they in the study have only mentioned 2 particular types of drugs, which was etoposide and panobinostat, where they studied the effect of these 2 drugs on the genes. So they observed that certain genes were upregulated, for example, LEPT1, BEX5, SAXO2. So there are certain genes whose functions are known, and there are certain genes whose functions are still not known. So it would be important for us to analyze the significance of these genes in cancer. So we can be looking at those types of biomarkers in our studies. Chandrani, can you move to the next slide?
Yeah, sure.
So basically what we can do is we can access different types of repositories that have already recorded data on significant biomarkers, which are associated with glioblastomas. So since glioblastomas are extremely heterogeneous, we would be able to retrieve data as to what kind of biomarkers are expressed in different tumor samples. And we will be able to correlate the data with the data that we have found in our paper. So once we get access to a good amount of significant data, which we will be doing data analysis on, we will be then able to compare the results of this study with results of other studies that have been carried out. The other thing that the paper has failed to do is to use more number of samples. Even though they had access to different types of tumors, they have used tumor samples only from a single patient. So if we are able to retrieve data from other patients and be able to analyze the effect of drugs on these patients as well, we will be adding to what the study has already done. So this is what we plan to do. Chandrani, I would like you to speak further.
Yeah, sure. So what eventually was the problem later on was they used the single cell RNA sequence pipeline that was there in the Tauber site was of drop seq capture method. So we wanted to reconfigure it for the microwell based capture method. And what they mentioned was the Illumina Nexic 500 runs, which we have thought to use and not use the other sequence runs that was there in the data set. Uh, uh, the Illumina Nexic 500 runs had 8 base index rate, and the 21 base read had 12 9 nucleotide length the cell identifying barcodes and a 9 nucleotide length UMIs. So that can be put in the pipeline, in the parameters of the pipeline. And they also did the gene expression analysis according to the marker genes that Pooja talked about. And also they used unsupervised clustering for visualizing the drug responses on the tumor sample, on the all the transform samples, and also differentiating the tumor and non-tumor populations. So these we will the analyzing these will all be in the single cell RNA sequencing pipeline, and we just have to remodel the pipeline somehow. And this is where we this is what they did for the first in the first analysis. They checked for the differential uh, differentially expressed genes for all the samples that were treated with all the 6 drugs. And they used different fold changes to generate the heat maps. And also in the clustering they did was the clustering they did below was they compared the control slices which were treated with DMSO, and they checked for the marker genes in the myeloid tumor and oligodendrocyte populations. And then they compared with the clustering that they did for the other samples, which were treated with the other 6 drugs. So this is the best portion of the analysis that they stopped. And then they went for analysis of the effect of panobinostat, like cell-specific responses of panobinostat and the cell-specific responses of etoposide. And and we want to check for the cell-specific responses of the other 4 drugs, which wasn't there in the publication. So this is how far we have come, and we still have to do the single cell RNA sequencing analysis. Yeah, that's all we have. Thank you.
All right, great. Thank you very much. I think very impressive presentation. What would be important to discuss here maybe briefly is the difference between the two single cell sequencing methods. In the DropSeq method, you actually have to have 2 files for each sample. And one contains the actual reads, the other one contains the barcode information. In microwell, you don't have that. You are assuming that the right types of data are in a single file. So there are some changes, like you said, to the pipeline. I think it would be great if you can again organize the data set that is available. I know that you showed kind of the NCBI screenshot where the bioproject is, but it would be great to see a table of specific samples, which what is their metadata, how exactly will you do the comparison that they did. So if you can work on that, and then I will also take a look if you could either post it on the forum for the intern program, or if you could have it in your project submission somehow, like a link to a Dropbox file or something like that. That would be great. So other than that, I think that, it's really good that both of you are working on this, and one person can focus on one aspect, another person can focus on another aspect. Typically that is how it works in real life. And I hope that anyone else in the program that might be listening in that could be interested in this project is going to be somewhat interested and potentially also join you as well. So we're going to focus on that for the next week, and really hoping that by next week you actually have the data. You can start seeing how those marker genes will help you identify different tissue or cell types, and start looking at the actual differences. But we do need to organize a lot better the data, to really see how the specific files are going to be used. Okay, so, but anyways, I think that's a really good start, and I'm glad that Chandrani, you've been working on this for some time now, and you have a good sense of the problem statement. Also, at the end, when you want to create a title for the project, think of a good title that really illustrates not just the type of data that you'll be using, but the specific question that you'll be asking. So what is the question that you're trying to answer? And try to make that question fairly narrow. Obviously, you need to give a broad background to others that have not seen or not, really heard a lot about glioblastoma or single cell RNA-seq. Those are good terms to use, but I think that it would be better if you could make a focused question and something that could be an anticipated result, right? So either a hypothesis, right? So our hypothesis is that the microenvironment plays a role in drug response. Or, if you think that it's more of an exploratory project, right? So focus on the specific comparison that you want to make, and then focus your results on achieving that one aspect of it. Because otherwise, you're just kind of doing a lot of different analyses and replicating what the authors did, and all of that is great, and you can improve and supplement, but ultimately you want to show some specific outcome at the end. What is the result? Where are you now? You're not going to necessarily solve the problem of glioblastoma in the next few months, but what is the specific contribution to the problem that you've identified will be from this project. Okay, so now I see that a number of the bioinformatics for infectious diseases participants are joining. We had a lot of good submissions. I was able to spend a little bit of time with some of our team members here at Pine to review some of the projects, explore some of the data sets, learn a little bit more about the reference publications that you have submitted. And I was able to email a few of you some of the feedback. So hopefully you had a chance to see that this morning and last night. I know that the most progress that I have seen so far was from Grace. If he is on here, Grace, I don't know if you have good internet access this time.
Hello, could you hear me?
Yes, I can. There's a little bit of an echo, but other than that, yes, we can hear you.
Okay, good. So should I present my? So I didn't
Yeah,
complete my work. I just did some um, field study and all. So these are the data set which I have so far.
Okay, so let me give you a couple of suggestions on how to present. It would be great if you start by saying a little bit about yourself. What is your background? What is your general kind of interest, maybe, or degree, etc. And then try to present this project, assuming that we don't have as much background in the project as you do. Take your
Okay.
time, don't rush through this. And then tell us kind of where you are and what your steps are after this.
Okay, I'm Vinod GV Aruraja, so from Sri Lanka, but I'm studying in Germany. So bioprocess informatics is my educational background. So apart from that, I have some medical biology background as well. So that's why I'm so interested in bioinformatics, especially in NSG, NGS analysis. So I found this course was so interesting for me. So I engaged myself during this lockdown. So I had to do something because I couldn't fly away, so I'm kind of locked down in Colombo. So I thought, okay, why shouldn't I try this? So I found some interesting topics and learned a lot of things within this month. So it was a really good process which I made. So with my background of knowledge, I tried to present something which I learned since last 2 months. So then I found a paper which was so interesting, like human bronchial organi. So which mimics the lung cell. So coronavirus is the currently hot topic that we were talking about and we were engaged. So I had a doubt, like during the transcriptomic course, I learned a lot of things about sequencing and reference genomes and all. So I had a doubt, like if human genome, different versions of human reference genome could impact the studies. So I found exactly a good topic which I could learn more. So I chose this topic. So these are the data set which I have found. So the scientists from Japan, they wanted to test whether human bronchial organoids, which is an advanced form of human bronchial epithelial cells, so whether it could be used as a in vitro test model. So what I found, two different sets, like two different doctors or PhD candidates. They did a experiment. So the first experiment was like they want to see the difference between or the similarity between the human bronchial epithelial cell and human bronchial organoids. So whether they mimic the exact human lungs, or at least do they mimic the do they have the important cell lines like goblet cells which secrete the mucin, or basal cell, or xylitate cell, or club cells. So in the first probe, I think the control is to A549 cells, which, as far as I know, kind of a tumor cell. So they found that HBC and HBO is expressing similarly, and then they thought, okay, we could process the experiment with human bronchial organoid. So in the second group of tests, they took the human bronchial organoids, yeah, and they have one set of control, and the other one is with SARS-CoV infected human bronchial organoids, and the third one is the infected cell, which was treated with chemostat, which is only permitted in Japan, as far as I know. So then I thought, okay, in the probe B, how could we confirm that the cells, the control cells, are not contaminated? So then I thought, like, why wouldn't I try with a metagenomic pipeline with the reference genome of all bacterial biomes? And I got the table, and with the help of W and the reference genome list from NCBI, I've managed to get a list of cell which was affected. And in transcriptomic part, I have planned to do RNA sequence pipeline. Then I will try to see the gene expression levels, and I'm going to compare it, or if I'm could I able to see some extreme upstream upstream expressions in any kind of cells. The cells which we concentrated about will we see at the last. So these are the two main pipelines I decided to do, like first pre-processing, then as they did, I will do the high set alignment, and after that I will run it through the HT sequencing. And I'm not sure about DC2 because I found that it's just some visualizing tool. Other than that, I didn't find anything important. I don't know, maybe I'm not sure. So then I managed to run the pipeline, but I think I made a mistake because I ran all 18 sample all together, and I tried to do the um, principal component analysis. So maybe it could affect the table. So, but I plan the next time I would do the probe of 1 as a first part and the probe of 2 as a second part, or else I might suggest to get the third table as well to visualize or analyze these part of area cells. So all these cells are human bronchial organoids which were treated and controlled and infected. But more other than that, both probes are not sequenced with the same equipment. Maybe it could also affect my result. So this was the metagenomics pipeline which I ran to see whether the viral infections are there. I mean the control test. So which I got the results were like that. So which I see some kind of expression wasn't there, but I'm not sure about the thing because I was kind of learning and doing. So I have to do it again properly, and I should see whether if I get the good result or is it repeating. So those are the things, the most important biological and medical key factors in my project I should see, or if I could see. So these proteins or the genetical region which is responsible for these proteins could be my important factors in my research, I guess. So that's it from my side.
Excellent. Thank you very much. I think this was a really nice presentation and
Thank
very
you.
detailed. So hopefully in your submission or on the forum, you could also, or maybe even in a direct message, you could send some of the pipelines that you ran. We can check, the different settings and outputs just to make sure that you're getting everything done correctly. What
Okay.
I didn't understand in your principal component analysis
Okay.
was the annotation between the two probes. So you said that you put all the files together and then you did PCA. And I didn't see in your annotation of samples which one belongs to which probe.
Okay. One second. So, I mean, in my pipeline, I put the probe 1 and probe 2 all together. So, and I grouped it into two different groups and ran the pipeline. I mean, this pipeline.
Yeah. But in slide 5, those are all of the samples, right? In the next slide?
This one?
No, in the PCA scatter plot.
Yeah.
Yeah.
These are, I mean, this is the cell line A59. This is from probe 1. So this one, and this one. These three are from probe 1, and these are from probe 2.
Okay.
So
Yeah.
on probe 1.
So that is an interesting fact because you have the biggest separation, right, is on x-axis. So
Yeah.
actually, your biggest separation overall is between the two probes.
Okay.
You know, typically, this is a good way to identify batch effects.
Yeah.
Batch effect means that, you know, if I do something here in my lab and you do something in your lab, but we're working with the exact same biological conditions and you get results that are significantly different from mine, you know, this could
Okay.
mean that maybe I have some contamination, or maybe, you know, my instrument isn't calibrated the same way. It's a very common issue with high throughput data. And a
Okay.
lot of times, you might want to use quantile normalization specifically
Yeah.
for that reason to make sure that the data is in comparable distributions for the significantly expressed genes.
Okay.
Uh, there's also a specific method called batch effect correction, but that's typically when you have more groups. For example, you have 3 different types of labs, and so
Okay.
you know, do multiple. So this is an interesting problem. However, I do recommend trying a number of different things here just for this scatter plot, doing
Okay.
quantile normalization between all of them, and also doing quantile normalization separately between the two
Groups.
batches, two groups.
Okay.
And then doing PCA also separately as well, just to see, you know, separately, right? In the first batch, you have a very nice separation between A549, the HBO, and whatever the third one,
Human
HP.
bronchial epithelial cell.
Yeah.
HP.
So, you know, so this is actually, you know, a good exercise to really understand what's in the data, right? If you have the same separation when you do those other, the second batch separately, and you have a, you know, good separation there, that would be at least biologically, you know, reasonable. The other part of this whole analysis, what I did not see, you
Okay.
mentioned originally in the first slide that one of your interests was in the differences between mapping on GRCH38 and
And
GRCH37,
it's
right?
37. I didn't run the 37 pipe line yet because there was an issue with which I
Indexing.
mentioned here.
Yeah, there was an issue with indexing, and I think that has been resolved. So
Okay,
maybe you
thank you.
can try starting that over.
Okay, I will try that.
All right, excellent. So, you know, I think overall this is a great start. You know, you already have a few slides. I think the background, at least to me, was fairly clear. You do present your methods. I think at the end, you want to, again, focus on what the main outcome of your study is, and make sure that that is clear when you have the visual, you know, very clearly there. So you just kind of summarize what you found so far.
Exactly.
Does anyone else have any comments on the presentation? Or also, I wonder, you know, when we started, I mentioned that the projects that some people have made already some progress on and have an interesting project coming together. So it would be great to see if anybody else is interested in joining, contributing, you know, making sure that everyone has something to work on. So just want to make sure that anyone looking at this might be interested in joining, or if anyone, okay, so Victor, for example, here is asking how to join this project. I think the best way is to say that you want to join the project, and then directly contact. Should I
I
call
can.
you Grace or Vinod?
Grace or Vino, whatever, however you feel comfortable.
Okay,
So,
so there you go. Victor is interested to join. Maybe Victor, you can post your email or you know, some
yeah,
way
that
to
would be
connect. And
nice.
okay, Okay, does anyone else have any comments or questions about this to Grace? Okay, excellent. Yeah, so I think this was a really nice presentation once again. Hope that you and Victor can connect together and just kind of bounce ideas off each other as you move forward with this project. And we're going to look forward to seeing this again. So thank you very much.
Thank you again.
Yes, Anthony would like to now share his progress. So let's do that. Anthony, do you want to share your screen and unmute your microphone?
Can you hear me?
Yes.
Awesome. Here, this is the first time I've actually shared my screen on Zoom, so let me just figure it out. Here we go. All right, cool. Can you see all this? My screen?
Mm-hmm.
All right, cool. So I don't know really where I stand right now overall with my
Okay,
project.
so 1 second before you actually start, maybe you can start with a little bit of background, who you are, what your general interests are, and then go into the project to give us some background. Not everyone
Yeah.
had a chance to learn about what you're trying to do.
Yeah, so I'm a student right now. I'm going into my 5th year at the University of Houston. And so I'm in the same situation that Grace mentioned. I got pretty bored during the lockdown, and this was an opportunity for me to pretty much do something new and also something applicable, into what we're dealing with right now with the coronavirus and everything. But yeah, so I'm studying mathematics right now and biology at the University of Houston. But this stuff, I pretty much learned this in my 4th year. Well, not all of this, but the principles underlying a lot of these things. So I thought it would be best if I tried to capitalize on those things and learn all of the hands-on techniques for actually making these phylogenetic trees, and also doing the proteomics analysis. I thought that was super cool using that Chimera program. So anyways, my family and I were all in the medical field. I've been working with them in their clinic for a long time, but we were still really confused on like how the coronavirus even like became such a problem, right? Um, and I actually wrote a paper about it for my class, and I learned that it's not the only coronavirus that exists. There's several other coronaviruses already in the world, and they all are in different hosts. And so immediately I wanted to start learning about how all of those coronaviruses are related, both genetically but also structurally. And I kind of took the approach of not just like, I didn't want to make a very detailed question from the start. I kind of wanted to just go and do the research and just kind of develop questions along the way, and then hopefully come up with like a answer, like a very specific question somewhere towards the end. But I basically broke this. I'm going to add a third part to this project, but this is the first part right here. The phylogenetic tree analysis. I'm going to redo this tree, by the way, because I found more hosts I wanted to add. So in this phylogenetic tree, I haven't put a legend anywhere yet, but it's color coded by organism, and I used the quasi-species phylogenetic tree method. So I did the quasi-species pipeline. I could pull it up right now, I guess. But I did the multiple sequence alignment for amino acids. And then, oh, and by the way, wait, no, these are all the, I don't have them listed on a PowerPoint yet. I'm in the middle of doing that right now. But these are all of the accession numbers and stuff that I used. I used 4 accession numbers per organism, and I tried to spread them out oh, it's been a while since I've been on here, so I guess it already unchecked which ones, but I have them somewhere else too. But pretty much each of these tabs in my browser is for an organism. And in each of these tabs, I downloaded 4 different accession number, or 4 different files that contain the amino acid sequence for the spike glycoprotein. Sorry about that. So I'm pretty much comparing the phylogenetic relationships for the spike glycoprotein for a bunch of different coronaviruses and different hosts. Sorry about that. My dog is super loud. Okay, so that's pretty much what underlies how I did the phylogenetic tree. So that's like the first part, and I don't want to go too in detail, but that's pretty much how that first part goes. And then I wanted to start looking at the protein structure of all of these different coronaviruses. So I went to the protein database here, and I found almost every host that I analyzed in the phylogenetic tree, but not all of them have the spike glycoprotein file available on the database. So I ended up having to, like for the canine, I analyzed dogs, their spike glycoprotein for the coronavirus in my phylogenetic tree. However, I could not find, I tried so hard, I could not find the spike glycoprotein or the whole protein in general for the, like the whole virus or anything like that in this website. So I ended up having to not include canines in my, I'll show you the part that I really like. This is the part that took me the longest amount of time. I spent most of my time doing stuff in Chimera. So this is all stuff I did in Chimera. On the left and the right, those are the same exact figures. Like they do the same thing, but I like the one on the right more because it emphasizes where the spike glycoprotein is, which is in tan. And the blue is the ACE2 receptor. So I'm pretty much showing how different coronaviruses bind in a similar fashion to this arm of the ACE2 receptor. And I already started making little figure legends for them. So like you could already see in panel A, that's the SARS-CoV-1 right here. Binding to the ACE2 receptor. The second one, the panel B, is SARS-CoV-2. Notice that they're very similar. And then for C, that's a bovine coronavirus. Notice that it's binding to the same region too. And then lastly, panel D is for civet coronaviruses. So if you're curious how I did this, I did it almost exactly what we did in session 8. I basically did the matchmaker program or command. So every single one of these coronaviruses have a receptor binding domain on their spike glycoprotein. So by the way, these are all spike glycoproteins. And I just did the matchmaker command for this exact this protein, and I matched them to this this whole entire ACE2 receptor, and it automatically would go to this this arm. I'm not really sure what the name of that would be, the binding region for that. So the reason why this middle figure is different is because not every single protein I found was in the correct conformation to bind. So I noticed that for like, so let's see this first one. So rhinolophus bat coronavirus. So I found a lot of bat spike glycoproteins on the protein database, but they were not in the conformation to bind. they would have a receptor binding domain, but it would not be in the up conformation. So I would have to, so I was like, well, crap, I didn't want to just like leave it alone. I still want to analyze it somehow. So I just matched the closed conformation of the bat coronavirus, which is in tan here. I used the same matchmaker command, and I matched it with the closed conformation for SARS-2, just so that you could see the structural homology between the bat coronavirus and this coronavirus we're dealing with today. So you can see that they're very similar, and that's probably why people also believe that the source of the coronavirus we're dealing with today came from a bat. So look how similar their proteins are, and that's crazy. So panel B, I did the same thing, and also in panel C. Panel B, let's see, that one was from a cat, I think, right? Yeah, feline infectious peritonitis virus. It is a type of coronavirus. It infects their intestines. So you can see that also looks very similar. This one has like more, I guess, longer branches out here. So this is like the view on the left is from the receptor binding domain. So down here, this is the receptor binding domain. And then if you just turn that, then this is the side view of the virus, and the receptor binding domain is on the bottom. And then that's the same thing down here. And then panel C, this is the avian coronavirus. So this is from a bird. And if you already noticed by now, all of these hosts I'm talking about are hosts that we see every day. We see, well, bats, you don't see them in the daytime, but they do exist in most cities. But birds for sure are a host we interact with every day. My cousin got bird poop on her recently, so there you go. That's a spillover event right there. And then we also have, you know, cat. This is the cat right here. And then, yeah, so civet is a type of cat, but yeah. And then bovine is a pig, and that's panel C, so yeah. But I mean, what I did on the left here is basically the same thing that I did on the right, where I'm just trying to show how it would bind. You could obviously see how the matchmaker command works. So this is the receptor binding domain that is matched to the SARS-2 receptor binding domain of the ACE2 receptor. It's the same exact process that Elia did in session 8. So one of the questions I was trying to answer is, how is the binding affinity different in all of these different virus receptor complexes on the right? And I don't really know how to do that type of analysis yet, but I'm guessing that, so here, let me just, well, no, I'm not going to do that. It's going to take too much time. But so every time you use the matchmaker command, you produce an output that is called the similarity index. And there's a bunch of other statistics that you create whenever you do this command. I still need to look into those things and see if I can somehow use that information to infer anything about binding affinity. But yeah, I mean, I feel like that would be hard to do definitively without any laboratory techniques, but still it might mean something if, yeah. So that's all I really have to say. But the third part of my project will most likely include multi-sequence alignment of amino acids so that I can start talking about which substitutions in the amino acid sequence affect overall where the virus binds, how, and if I can find anything on binding affinity, how might the amino acid differences affect the binding affinity. But that might require me to like maybe read a different paper that talks about binding affinity, and then look at multi-sequence alignment. But if anyone wants to join me at this stage and help with the multi-sequence alignment part, feel free. I've been doing this all by myself, so I really would like some help if anyone is still searching for a project to join. And that's all I have, Elia.
All right, thank you, Anthony. I think a very nice presentation. It's clear what you're trying to do. I think there are a few questions that I would have. So you are showing here different ACE or different proteins from the virus. And so there are many different types of viruses that are known to be specific to those hosts. Are you also looking at the variations in the ACE2 protein from these different hosts?
I actually did not. I only compared, so I used one type of ACE receptor the whole time. I didn't really, that's a good question though. I thought about doing that and taking the ACE receptor from each organism, but I thought about it and I was like, well, if I want to like try to study how like a virus might affect a human, so like I'm basically testing the hypothesis of is it likely that a virus from a bird will infect a human and matter? Like, will the binding affinity be high enough so that it will be considered infectious and it will spread in the body? So like in every single one of these panels, I use the same exact ACE2 receptor. It's the same file that you used, I think, in your session 8. I did that mainly also just to be consistent, so that I knew what, like I wanted to make sure I controlled for which receptor I was binding each of these to so that I could compare their binding affinities without any, you know, confounding variables.
Yeah,
But yeah,
no,
I mean,
that
I
makes
think,
sense. So I just wanted to make sure that when you're, comparing how it binds, you had a specific reason for why you were doing that. So I think that's great.
yeah.
The other thing that I wanted to mention, so there's a comment from Mohit, who has a PhD in chemoinformatics and has a lot of experience there. So he's suggesting some methods. So I,
Hmm.
suggest you also reach out to him or look at some of these methods.
That'd be great.
The other question that I would have is about the, looking at how the viral protein, right? So you're talking about different conformations. So that's one, obviously in literature, there's a lot of reviews about the different environments. For example, you're talking about, some of these viruses affect different organs. And so in the gut, the environment will be very different from the lungs, would be very different from, epithelial cells in the upper respiratory region or deep inside the lungs. So those different environments actually cause different states for these proteins. The proteins can change shapes. And so when you're looking at this kind of an analysis from purely structural perspective, it is extremely difficult to conclusively, like you mentioned, identify how that impacts binding at the structural level only. So what you have to take into consideration are the specific regions, those amino acids or even nucleotide sequences that might produce some change in the physical chemical properties. There's a great review paper I'd love to share with you so that you can take a look at what exactly they did from a purely in silico approach. And then also how you plan to, look at the probability. So let's say the affinity is at a certain threshold, right? How would you actually conclude from whatever the change is, right? Whatever the change in that affinity is, or how likely it is for that protein to still be able to bind? How would you actually take that to the next level and say, based on this data or based on this analysis, this is what my conclusion is?
Yeah, yeah, I see what you're saying. Because Yeah, right now I just finished doing all of these things, and I'm still trying to answer that question of, yeah, like how can I infer what binding affinity looks like using what data is given. So while I'm sharing my screen, I'll just show you what I'm looking at whenever I mention like all of those statistics.
Yeah, I know of the statistics that you're mentioning, and those are great statistics to have, you know, to just have a general sense of difference, right? So for example, it will say, you know, it went from 90% to, you know, let's say 80% or something like this, right? So that's one side of it. The reason why I originally mentioned looking at ACE2 receptors from different organisms is because it's not like we have no idea of a virus actually coming in from a different host, right? That's a common thing that happens. We have the avian flu, we have all these different coronaviruses, right? So that's something that happens all the time. We know that that exists. Once a virus, like the novel coronavirus, has actually infected the human population, right? One of the questions is whether that same virus can then spread just between people, or could it infect, for example, pets at home, the human virus infecting pets at home, and then those pets becoming, you know, they could be asymptomatic, or they could even have some mild condition, right? They might feel a little bit sick, but we're not going to know because they don't talk to us, they don't tell us. And, you know, and then they become the ones that are spreading the virus, right? Or for example, if we're talking about, you know, meat consumption or something like that. So that's why I think looking at ACE2 receptors from other hosts might be interesting in the context of your question.
Yeah.
And, you know, the differences in, let's say you take a feline ACE2 receptor and you have a feline coronavirus. Well, just like a feline coronavirus could infect a human, you know, novel coronavirus could infect the feline, and those are interchangeable comparisons. And that's why I think at this point, as you're starting to map out what kind of data is available, what types of analyses could potentially be done, and you've properly identified, I think, you know, the phylogenetic relationships and the multiple sequence alignment and looking at specific regions and then testing what happens at the structural level. Now I think you're at a great place to really define that question more specifically and to say that, you know, this is how this type of analysis can actually answer maybe the whole question or maybe just a part of the question that opens up, you know, the next question that you think should be addressed as more data becomes available.
Yeah, yeah, I totally agree with that, and I need to start reading up on more articles. So far, I've only been reading the same one, which is this one. I think you shared it with us. It has this figure right here, and I thought about looking at and trying to recreate this type of figure too, to show which type of amino acids are essential in the binding. Um, I'm not really sure how to make all of the different things they have here. Like they show the interactions between amino acids, like with these dots and stuff. But yeah, no, I understand what you're saying, and I totally agree that I need to start paying attention to also the environment of what these virus particles are in, because like I said earlier, that the feline coronavirus is in their intestines, or at least the protein that I found right here in panel B. So that's a low pH environment, clearly, and so that would be something that I might need to take into account. Because I think in a different, so like whenever I looked at SARS coronavirus, uh, this one in particular, the one where I bind the SARS spike glycoprotein to the ACE2 receptor, it's in a low pH environment on purpose so that this receptor binding domain goes in the up conformation. However, in panel B, it's also in a low pH environment. I don't know how low, but it's in the intestines. Well, that might not actually be that low of a pH because it's not the stomach, but still there's
Right. So Anthony, I think, you know, again, to leave some more time for the others to also share, I think you're on to, you know, a lot of great insights from what you've already done. And, you know, I think it would be very nice that between now and next week, you're able to first, before you do any additional kind of analyses, right? Like you said, read up on some literature, try to identify the specific series of steps that would be designed to answer a particular question. And you'll see how, you know, significance of the different environments, of the different hosts, of the different ways that the data was produced will help you eliminate any variables that you're not controlling for, and you know, really see whether that could answer the question that you're posing. And so what's, you know, a little bit kind of unclear is what is that specific question. So I do want to say that if you look in the chat, you have messages from Mohit there, and you have messages from Urja, who is talking about Pymol, which is a very similar software to Chimera. In Chimera, there's a lot of methods also that you can use to create the figures that you were showing in the publication, and also measure affinity and things like that. You can also see Chandrani here added about molecular docking and energy minimization. So I feel like there are a lot of technical methods that are available for you to get to the right kinds of conclusions if you are designing your analysis in such a way that, you know, it answers a specific question.
Yeah, no, yeah, I see that. There's a lot of different things here. Yeah, thank you so much. I don't want to take too much time away from other people, but yeah, thank you.
All right, great. Well, thank you, Anthony. And again, like Anthony mentioned, he's open to anyone else interested to contribute to his project. If you would like to contribute to his project, Anthony, you might want to put your email address or post that on the forum or something like that so people can reach out. I'm sure that others can contribute here as well. Okay, so I think that, you know, these two projects, again, we saw a presentation from Grace, we saw a presentation from Anthony. Before that, we had a presentation from Chandrani and Urja that are going to be working on their project in glioblastoma. I wonder if anyone wants to volunteer to go next and present any progress that they have on their project. And Anthony, maybe you can in the meantime stop sharing the screen. So, okay, thank you. Okay, so Sithira and Misaka want to present next.
Hello?
Yes, hello.
One second. Share screen. What? One second. Hello?
Yes.
Yeah, I can hear it. Hi, hello.
Yeah.
I'm Sen Nayak. I'm Sithira. I'm a general physician based in Sri Lanka, and Sithira is also a doctor. So we are quite new to this bioinformatics game. So it's kind of how Vinod said that during the pandemic time, we were quite interested on discovering how all these researchers publish their and give their opinions about viral strains. So we just happened to come across your bioinformatics course, so we just joined up. So our problem statement. So we thought about several projects, and then we settled on dengue because dengue is very endemic to Sri Lanka. It's actually an epidemic, and it has spread to most of the countries now. But it's quite important in a Sri Lankan context because of the monsoons and everything. So the problem statement is a study of a complete dengue viral genome available from Sri Lanka and comparison with dengue hemorrhagic fever epidemics. So what we wanted to do was to compare pre-epidemic, because I found a paper saying there were certain timelines where dengue hemorrhagic fever epidemics happened in Sri Lanka and all over the world. So to isolate that, we wanted to take some viral strains that has been sequenced prior to that and then compare it with epidemic. So it's clearly the samples are not tagged as epidemic as in like DHF, but the years match according to that, like 1989 in Sri Lanka, 1996 in India, and Philippines it was 1956. So at the beginning, so because we are quite new to this, I think our project, we were afraid that it will just be beyond our scope and beyond the constraint of this course. So we try to contract it to something that is doable and achievable during this timeline. So we just started in this project, we will be completing the initial portion to identify variations to comparison of Sri Lanka's dengue viral strain 3. So what we will do is take dengue viral strain 3 and compare the strains which are available from Sri Lanka. It's not a big amount because Sri Lanka doesn't do extensive genome sequencing because we are not a rich country.
Yeah,
so we
there
have
are 14 strains, completed 14 strains in NCBI.
Yeah, there are only 28 from Sri Lanka, and then only 14 of those are on dengue 3. So what we wanted to see was the correlation between if there was a genetic
component,
genetic component and any variation that would contribute in the increase of dengue hemorrhagic fever patients. So I'll just give a brief introduction about dengue fever for those who don't know. So dengue is a mosquito-borne disease now which is prevalent throughout the world, and it is very important in Sri Lanka being a tropical country. So I just explained why it was very important because of past literature mentioning about certain epidemics happening in certain eras and then continuing till today. So brief introduction on this. The dengue virus is a flavivirus, is a single-stranded RNA virus with 4 serotypes, which is dengue 1 to 4. So, and dengue 1 and 2 were the predominant ones some time ago, maybe in the 1960s, 1950s, and then dengue 3 came out and then it was more virulent than the others. And dengue 4 is mostly connected with secondary infections. That means people who get dengue first time and then second time they get it again, it's much more severe. And the dengue virus itself, the genome is 10,500 to 11,000 bases long, and then divided into a structured and an unstructured portion. The non-structured portion is the core, the membrane, and the envelope, and the non-structured portion is the more familiar NS1, and then those are non-familiar to most people are NS2A, B, and then N3 up to NS5. And the envelope protein is the main biological function and the virus. So I think this is, I'll leave the PPT later, y'all can just read on those things. So vector transmission is mostly happening through the mosquitoes, which is from the Aedes aegypti, but it can happen with other species as in Aedes albopictus and Aedes coligenesis according to the situation, okay? So pathogenesis is not known too much about this. That's the main question we have in dengue because we don't know how it really happens. So it's fully understood. So there are 2 main hypotheses behind it. One is the secondary infection or immune enhancement hypothesis. And the other one is a hypothesis assumes that dengue virus vary and some are associated with high virulence, severe disease. So this is the second hypothesis or idea, pathogenesis idea is what we are looking at and what we have touched upon. So clinical manifestations, something that people don't know about dengue is everybody thinks dengue fever is dengue hemorrhagic fever because that's the ultimate outcome. So that's not the case. Most of the dengue virus patients will have undifferentiated fever or asymptomatic and something we call without hemorrhage dengue fever. Okay, so most of them will have a platelet drop and they might be mild cases. What we always worry about is this portion which is with hemorrhage and the shock portion, which people always think that is the ultimate result of all dengue cases, which is not the case. So I'll skip this. Yeah, so this is the epidemiology data from 1954 to 1980 and stuff like that, which I already explained. So what we are going to do is taking the 14 samples of dengue 3 available from Sri Lanka. We did a multiple sequence alignment to compare all the strains, and we created a evolutionary phylogenetic tree to visualize, to see which strains were related. And then what we wanted to do was compute all the mutations and the amino acid changes and allocate them into the structural and non-structural domains in the viral genome. And then our ultimate goal, which I think is beyond the scope of this course, is to see if those changes resulted in any pathogenicity, increased pathogenicity or virulence of the,
which leads to a dengue hemorrhagic fever.
Yeah, dengue hemorrhagic fever, increased cases in epidemics, which would result in, as in like it will stimulate the immune response of the patients and make it more virulent. And a large portion of dengue patients will transit into dengue hemorrhagic phase, which is leakage phase. So this is just a picture of the MSA pipeline we ran on the T-BIOS system. We have been also using several other softwares because we are new to this. So we are trying to figure out ways of computing and getting all the statistical data out. We were not very familiar with. So we use Mega, and we are also trying with Eugene. Mega was more user friendly, and also did some blast on NCBI to get comparisons between the viral stains on percentage of likeliness. So this is
MSA
preliminary.
The MSA first.
Pardon?
The MSA.
Where is it?
Yes. 1 second.
And so are you showing in this table right here, you're showing number of mutations in each protein?
Yeah, we'll get to the MSA. So what we did was on Mega, we kind of calculated between all the strains, from compared it with each other. So this is a number of nucleotide variations between each strain. So I put it in the graphical form, so it's easy to read. So the top one is the reference genome, which is the NC001472, but it doesn't have a collection date, but it's most likely around close to year 2000. But what we wanted to do is we use this one, dengue 1983, which was pre-epidemic strain, and then compared it with the others, and you can see that most of them have high mutation rates compared to with the reference to 1983. So we also ran a phylogenetic tree which shows that this 1983 sample is different from the other strains.
Also, we ran a blast on the 14 strains as well. Where was that? That was, yeah. So this is the blast that we ran that compared the different strains. So as you can see, was this the correct one?
Yeah. No, 76.
Oh. Can you see our screen? Hello?
Yeah. So right now, Misaka, I believe, is sharing his screen. So he would have to stop sharing his screen for you to.
Oh, we're in the same. We're in the same place.
We're in the same place. I'm not sharing anything. We are in the same. We are using Citara's computer.
All right. So which one was 1983? This one, right?
Yeah, this one. Last one.
Why is it?
I don't click on it.
So this we compared the other 13 strains against this strain, the KF95, because this was a pre pre-epidemic strains that we could find. So you could see the wait, is this the correct one?
Yeah. So here it shows, now we compared it against the top one, which is a 1981 sample. So the largest difference shows against KF21. If we flip it and make the KF 955476 strain the primary one, then it will show as 100%, and everything else has 95 showing the difference between those strains against that. So what we wanted,
you might be showing something, but I'm not sure that we're seeing what you're looking at. We are seeing your phylogenetic tree in PowerPoint.
Oh, okay. We are looking at an Excel.
One second.
Wait, stop and show.
Can you see Excel sheet?
Yes, now I can see that. Yep.
Yeah. So the bottom one was the pre-epidemic. There are other strains which are also pre-epidemic. So the word epidemic is mentioned in a paper. So that just says that as an increase of dengue hemorrhagic patients, which has transited into leakage phase, right, from normal innocent dengue fever. So this strain shows 95% on blast. So if we flip it and make it the reference, it would show as 100% like the top one, and everything else would be close to 95. So it shows as similar to the evolutionary tree that
there is a difference between.
There's a significant difference. There's an increased mutation rate of dengue, and there's a significant difference we can locate on the genomes.
From 1983 onwards, that's like a 5% difference.
Yeah, and there might be more on later strains. So what we wanted to do was we found a paper, this one, right? No.
Go ahead.
We found a paper which was similar to what we wanted to replicate, which an Indian team had done, which had used the dengue 3 virus from Rajasthan, because Rajasthan never had a sequenced dengue samples. They did that and did the analysis on it, and got this table where they mapped all the mutations and the positions in reference with the non-structural and the structural portions of the protein, the DNA, the viral genome. So according to this, we could identify, or even later on, to see which mutations resulted in amino acid changes and then changes in proteins, which could have stimulated the human immune system differently and changing the immune response towards dengue. Maybe certain, for example, last time I also mentioned like NS5 has different immunostimulatory components where it might stimulate cytokinins and interferon. So that causes vascular permeability and increased leakage. So if we can pinpoint if these mutations increase, or during the epidemic it has increased the number of patients going into leakage, then we can also see, we can use precision medicine or maybe other methods to use that to isolate or even identify medications, how to combat dengue in coming up with a
last question.
Possible treatment method. At the moment, we don't have definitive treatment for dengue. It's all supportive fluid resuscitation. So these are the questions that we have and having problems with right now, which Ilya, you might have to help us with. So we have a problem with computation and accumulation of data, because we are quite new to this, actually brand new. We don't know if those softwares offer these kind of things, or we do have to do everything manually and count them.
Yeah, because the MSA file, we have everything with the variations, but we need to get, we can calculate how many variations are there, but we don't know what position it is. I mean, we can look at it manually, but that's going to take a long time.
Yeah, it's similar to that PDF file, the study that I showed. It has shown all the mutations, their positions, right? So we can correlate it to the specific structural and non-structural portions depending on the genomic position.
So like this.
Yeah, what we want to do is build this.
And also, if at all possible, we would like to know, is there any way to figure out how these variations, do these variations cause any what changes it cause in the protein's function? If at all, if that's possible to find out. So we are at this the place we're at right now.
Okay, well, I thank you for the presentation. I think it gives a very good overview of the problem and where you're going with your analysis. All of these are definitely possible. So, and that's what we would like to get to by next time. What would be important is, as you can see, you've got 22 slides here, 21 slides, which is a lot of information, and it's good that you're assembling it all here. What is missing from this information for me to really help you out are the specific pipelines that you ran, organizing your samples with the metadata so that you can say in maybe similar to what Grace did before. As you saw, he had specific samples and placed into groups. So it's very clear in a single slide just to understand these are the data sources, this is what why we're using them, and this is how we are using them.
So you have the accession numbers that we are using over here. So
yeah, which is great, right? But I still don't know from this table and the other tables that you've shown, or the other, let's say, phylogenetic trees that you've shown, how do they relate to each other, right? So what I would like to just maybe, it would help you to have like a single slide where you just have, okay, these are the samples that we have. This is why we are doing this comparison. And these are the specific changes that we're looking for. Just even schematically represent your analysis schematically.
All right.
So then after you do that, and you've already done a lot of analysis, you know, basically what you're doing is a lot of multiple sequence alignment and phylogenetic tree. So all of those outputs, there's a pipeline that produces a single output that is directly imported into SeqNav so that you can do all of the visualization. So that's why it would be helpful for you to reproduce that pipeline. And once I see exactly the files that you're working with, you know, I'm happy to help with that as well. The final thing I want to say, you know, you're mentioning how these variations that you're finding at the genomic level, how do they actually correlate with the disease manifestations in patients? Asymptomatic, you know, mild, or hemorrhagic fever, right?
Yeah.
And you kind of know in a general sense, because of the years, what happened in those years. But you don't have any specific samples from patients. So I think there are some patient samples that you can actually find on SRA. And what would be interesting, I think, and I'll just give you an example, I'll put it here in chat. I think what would be interesting is once you find the specific variations or specific strains that are more associated with, you know, to explore what are they associated with, is to have some patient samples where you can detect the same strain and see how similar or different it is from what you found. So one part of the interpretation is annotation, like what you've described, right? This protein is responsible for that, and we know it from certain studies, so we can look at mutations in that protein and kind of think that it is going to cause some change. Another direction is to say, okay, if this is found in a patient with a hemorrhagic fever, but is not found in a patient that has mild symptoms, then I know that that mutation actually plays a role in causing that hemorrhagic fever. That would be one of the possible reasons. There might be other reasons, right? For example, the immune condition, or, you know, other comorbidities, or many other factors. And as many of those factors we can eliminate from the study, the more conclusive your results are going to be.
Yes.
Okay,
but,
so yeah.
One problem that we do have is, like, from, you know, if we can expand it into other countries, then we have a larger data set. and maybe different samples that were specifically taken from dengue hemorrhagic fever, and then non-dengue hemorrhagic fever patients. But on a Sri Lankan context, because we wanted to contract this into something achievable at this timeline, so we contracted it to Sri Lanka. Sri Lanka has very few samples, only 28, and even India has a very small percentage of samples, which really don't tell which the state of the patient, if it was dengue or dengue hemorrhagic, because sampling like that does not happen in Sri Lanka. I mean, maybe it happens specifically in a research institute in Sri Lanka, but none that I'm aware of right now.
Right, and so unfortunately, you cannot answer. Unfortunately, you cannot answer every question that you have, right? If you don't have the data. So that's just the reality of life. And you know what our purpose here is to identify data sets that can answer a specific question. So if from the start you know that the question is not possible to answer given the limitations of data that you have access to, that's just a dead end. And if you would like to find creative solutions on how to overcome that, I think that's also possible. For example, you can say, this is the data that we have from Sri Lanka. This is what we were able to identify, specific mutations in these positions. However, we also have some other data from other countries. We have a lot more information on patients. We have a lot more information on maybe diversity of viral genomes inside patients, right? There's a lot of other components to virulence. And then you can infer by similarity what these ones that you have from Sri Lanka are most closely related to.
It's an inference that we can make because nothing on dengue is conclusive at the moment. So it's an inference for further research and maybe a preliminary data collection, which is what we are doing. So,
and also, I think it would be great if, as a result of this, right, your conclusion, like for example, you're saying there are specific research institutes that deal with this and have the capability to generate data. If your study helped define what specific types of data can conclusively answer a specific question that is important, you know, that would be a great outcome as well, right? You know, you've done the research, you've done the comparison, you've identified this missing part of the data that you need. You know, that's a grant proposal, right? That right there is a proposal for, you know, a justified use of money to answer a specific question that is important to your country or to your community.
Yes, I think we are going with that, maybe, but that's beyond our scope. But
currently, these are the two main problems
because
we have.
Genomic sequencing and comparison of dengue viral genomics is not done. I mean, it's very scarce.
In Sri Lanka.
In Sri Lanka, specifically in Sri Lanka, because we don't have the resources to do that. And
okay, so you know what I suggest, again, we're running a little bit over time, so what I suggest is, you know, this was a great presentation on where you are. We'll work with you to get you much farther along by next time. I want to briefly ask if so there's two of you, right, working on this project right now. Are you open to anyone else if they want to participate and help out in any way?
Yeah, we are. Yes, we are.
Okay, so if anyone else looking at this study is interested, you know, they've done a lot of work already, have collected a lot of data sets, have a lot of insights into dengue disease progression. Maybe you can put your email address or the way to contact you. And if anyone is interested, you can reach out to them. We will, by next week, so just to summarize, we heard today at least 4 different project presentations. Thank you again for, you know, putting together the slides and organizing all this information. I think the presentation is great overall. Our goal by next time is to allow the ones that were not able to present today to either pick a project that they would like to contribute to, or present a project of your own. To be able to present, I would ask that, you know, today we saw great examples of how you can organize your presentation or start organizing your presentation. Please share your slides ahead of time so that we can review and take a look at those slides and maybe help make the number of slides, not more than 10 for sure, because we want to fit in, there's at least, I think maybe 10 or 12 other projects there. Babis is asking here if I've received the project. Yes, I actually replied to both of you this morning about the project. It would be great to see a few slides, just like you've seen today, so that we can get a sense of where the project is. And so let's try to work on this for next time. Have at minimum 3 slides, at maximum 10 slides. Follow kind of the specific objectives that were in the form. What is the problem that you're trying to solve? What is the data set? How can you clarify, what type of data this is, and what does this data represent? What types of conditions, what types of comparisons that you're trying to make? If you can make sure that you then already tried doing some of those steps. So what we see today is a lot of analysis that was already done. And to describe the analysis, you need to actually show us, maybe the pipeline, maybe have a link to that pipeline so we can take a look if something didn't work well. Probably you need to organize your project plan into some kind of a flow chart. I collected the data, I did exploratory analysis. Remember, we went through that kind of a chart, right? Exploratory analysis, I generated this hypothesis. Yes, you can definitely start running the pipelines. And if you need help finding the data and uploading the data, you can reach out to us. We'll help you upload the data or make sure that you're getting the data, the correct data sets uploaded and things like that. And once you've got some idea of what your hypothesis is, try to, again, make sure that the hypothesis can actually be answered with the type of data that you have access to and with the type of analysis that you're going to do, right? What is an answer and what is not an answer, right? So if you find a data set that, for example, describes gene expression, and you're trying to analyze genomic sequences of viruses, how do you actually identify that genomic sequence in whatever RNA-seq data you have, right? So those would be important things that we're going to be looking for. Also, Claudia is asking here, please, I need your help. Can you answer my last question? I just posted in the forum. I haven't been able to upload any data set into my pipeline. Don't know what am I doing wrong. Yeah, so those are the types of questions. If you can put them up on the forum, I will definitely be looking at those. And after next session, right? So if you don't have a presentation by next session, you haven't joined anybody else's group, then unfortunately, you're not going to be participating in the project presentations. We'll have a few experts that have backgrounds in genomics and bioinformatics, and they will provide you with feedback on the projects that are actually ready to be prepared, right? So what we saw today are drafts. Hopefully, they are in much better condition by next time. They need to be concise. You're not going to have a long time to present. They need to be concise. They need to be well organized so that it's clear and the questions are addressed in your presentation. And they have to, obviously, by the time that you're showing them, they have to have all the information that was required in the project submission form. Okay, so again, thank you everyone for joining today. I enjoyed looking at your projects that you submitted by forms and researching some of your data sets that you've posted there. I did mention that, yes, you can start the analysis, but again, look at my comments on the data sets that you've selected. Specifically, Athanasios, you did select there a lot of different bio projects, a lot of different single cell data sets. I think before you jump into the analysis, let's define a little bit better what kinds of comparisons and what kinds of specific files from those you will use. Otherwise, it's just going to take a very long time, and it's a little bit unclear on how the annotation is going to happen. I don't know if you had a chance to see the presentation today that Chandrani and Urja made about their single cell data and some of the problems they ran into there. But those are some of the things that we will try to address in the next week or so. So yes, you can share your presentation. The best way to share your presentation is if you have a dynamic presentation. Dynamic presentation could be in Google Slides. There's a lot of many other options. Basically, what that means is you have one link. You can have Google Docs or anything like that, right? A Dropbox link, something that you change, and we can continuously go back to that link and see the updates and see the changes that you're making. So you don't have to every time upload new files and send new files by email, right? That way we will lose track of where we are with the different projects. So that's why the form that we gave you is also dynamic, right? If you make changes in that form, we only see your latest submission. Okay, where could you upload the PPT? If you have a Google account on Google Drive, you can upload your presentation on Google Drive. If you want to make it in Google Slides, that's even better. That way we can comment directly right there. If you want to use Dropbox, you can use Dropbox. If you don't use any of those, we'll email you some options so you can register for free and use some kind of an sharing link so that we can take a look. All right, great. So thank you again. Yes, and Bibsha here mentioned the emails. So once again, by next time, not less than 3 slides, not more than 10 slides. Concise and focused on specific types of analyses, on specific types of data sets. You don't have to focus right now on results. Just make sure that the analysis is explained well, data sets are explained well, and you have a clear sense of where you're going with this. After that, we will obviously work with you on making sure that the analysis is already complete and it's done. And that's the last part of your project, is you're going to be working on interpretation. What are the findings that you have and what does it mean? And so then your ultimate goal at the end of this is to have a presentation that has no more than 10 slides, and you have some kind of a single slide, like a graphical abstract, that explains your whole project altogether, including methods, data sets, findings, all of those things, future directions. You can think of it as a poster presentation that you would present at a conference. Okay, again, thank you all for joining today. Sorry for those that did not have time to present. For those of you that did present, you did a great job, and we'll see you again next time.